William Paley was an English Christian apologist and philosopher. He is best known for his natural theology exposition of the teleological argument for the existence of God, which made use of the watchmaker analogy. He believed that the complexity and perfection of nature were powerful arguments for the existence and attributes of God. Paley famously made an argument by analogy of organisms as watches and of God as a watchmaker, according to which complex structures, like a watch, could not emerge accidentally but require the existence of a rational designer, in this case a watchmaker. Paley's watchmaker analogy is a classic. Paley writes, "In crossing a heath, suppose I pitched my foot against a stone and were asked how the stone came to be there. I might possibly answer that for anything I knew to the contrary, it had lain there forever. Nor would it perhaps be very easy to shew the absurdity of this answer. But suppose I had found a watch upon the ground, and it should be inquired how the watch happened to be in that place. I should hardly think of the answer which I had given before, that for anything I knew, the watch might have always been there. Yet why should not this answer serve for the watch as well for the stone? Why is it not admissible in the second case as in the first? For this reason, and for no other, that when we come to inspect the watch, we perceive what we could not discover in the stone, that its several parts are framed and put together for a purpose, that they are so formed and adjusted as to produce motion, and that motion so regulated as to point out the hour of the day, that if the several parts had been differently shaped from what they are, of a different size from what they are, or placed after any other manner, or in any other order than that in which they are placed, either no motion at all would have been carried on in the machine, or none which would have answered the use that is now served by it." Without knowing about biology as we do today, Paley made observations which are spot on and have astounding significance and correctness applied to the reality of molecular biochemistry. Let's list the points he mentioned again. If the parts are differently shaped, differently sized, placed after any other manner, or in any other order, no motion would be the result. The individual parts of the watch must be designed in order to be shaped and sized correctly, so they require an intelligent designer. Each part must be complementary in order to be able to be placed at the right place, and the assembly also requires the right sequence. The same is the case in a multicellular organism. This principle is called structural complementarity and is the very essence of biomolecular recognition. In the same sense as in a watch, one part like an apoprotein without its substrate or prosthetic group must precisely fit and be able to interact with other parts. In biology, structural complementarity is the significant clue to understand the fundamental properties of biological systems. Biological systems, from the macromolecular level to the cellular level, operate via specific molecular recognition mechanisms based on structural complementarity. A sperm, for example, recognizes an egg. All these interactions involve structural complementarity between molecules. So, according to the modern evolutionary claims, the watchmaker argument has been refuted by the theory of evolution. Richard Dawkins borrowed the name from Paley for the title of his 1986 book, The Blind Watchmaker, which attacked the idea that complex life needed a religious explanation. And still today, when the watchmaker analogy is invoked, the immediate answer is the analogy has been refuted through evolution by natural selection. But has it? The emergence of multicellularity was ostensibly a major evolutionary leap. Indeed, most biologists consider it one of the most significant transitions in the evolutionary history on Earth's inhabitants. We will address this question by giving at what hurdles unicellular organisms would have had to overcome in order to evolve into multicellular organisms. How do changes in development create new body architectures? Are heritable changes enough given the constraints imposed by the necessity of organisms to survive as it develops? These questions are critical in molecular biology, physiology, cell biology, genetics, anatomy, cancer research, neurobiology, immunology, ecology, and evolutionary biology. The study of development has become essential for understanding all other areas of biology. In turn, the many advances of molecular biology have finally given elucidating answers. Have they been predicted correctly and in a satisfactory manner by Darwin's theory of evolution? The advance of science has it made possible to solve the riddle. The black box has been opened. Has the evidence led to confirm evolutionary predictions, or have they been falsified? Evolution would have had to go in a gradual, slow, increasing manner from one eukaryotic cell to an organism with 2 cells and so on, to get into the end an organism with millions, billions, or even trillions of precisely differentiated cells like the human body with 37 trillion cells. Let's suppose there were unicellular organisms, eukaryotic so-called protozoans, and evolutionary pressure to go from 1 to 2 cells. What and how many mutations would be required in the genome and have to be selected and fixed? Mutations would have to provide the selection of a considerable number of new internal cell functions and created new information for these to emerge. There would have to be set up a way to divide the tasks and assignments of cooperation to consume energy, survive, and reproduce. But how could just this apparently tiny, but in reality huge step in the molecular world have occurred? And why should it have? How did cells supposedly make the transition from life as single cells to split tasks, set up the information to start existing as bicellular organisms, and associate and cooperate as teams such that they work as a single cohesive unit? The change would englobe the modification of shape, divide functions, and assign to each cell special tasks, structure, and entirely new features of development, ways to connect and stay connected to each other, communicate, and cooperate in order to start doing things in a combined manner. And all this transition would have to be advantageous during each transitional step, resulting in an organism having an architecture where specialization of each cell in their functions would provide an advantage of survival. If they would duplicate and stick together without specializing, they would just remain a group of cells or a colony. Evolutionary literature claims that such transitions supposedly occurred 25 times independently, which is called convergent evolution, since cell-to-cell adhesion and communication is achieved in various ways in plants, animals, fungi, and algae. As evidence reveals, a considerable number of innovations would have to be implemented as listed above. The technical demands of multicellularity are extraordinary. Cells that commit to living together need a whole new set of tools. They have to come up with ways of sticking together, communicating, and sharing oxygen and food. They also need a master development program, a way to direct specific cells to take on specialized jobs in different parts of the body. And a self-destruction program is necessary. The process is called apoptosis. The genes participating in adhesion in animals might have been extant in the unicellular ancestor of animals. Most of the domains typically found in animals are present, for example, in choanoflagellates. However, the function of such a diverse set of adhesion molecules in a unicellular organism is not yet known. There are more demands and requirements which will not be elucidated in this video. Why did cells not remain single-celled? In Astrobiology magazine, the answer of this question is usually cooperation. Our cells benefited from more working together than they would from living alone. Really? An article in Science magazine in June 2018 admitted that the transition to multicellular life as momentous as any in the history of life. Until recently we had no idea how it happened. The gulf between unicellular and multicellular life seems almost unbreachable, so they confessed. If so, does the explanation above not seem, let's say, a little bit simplistic and naive, or worse, even pseudoscientific? In the paper The Origins of Multicellularity, the author writes, "My line of reasoning is that the first step in the evolution of multicellularity was a size increase due to an accident. In other words, a mutation that prevents the daughter cells from separating. If the larger cell mass has any advantages, such as ensuring the safety of the germline by producing a protected internal environment, then natural selection will see to it that the novelty is retained." Now, cells require ultra-sophisticated proteins that keep them together. Arguing that an accident did hold them somehow together seems nothing more than just so guesswork. We will give now a closer look at the mentioned innovations. Cell differentiation. That is already a huge venture. Stem cells can generate transit cells and finally differentiated cells. In most cases, stem cells can produce more than one type of differentiated progeny, multi or totipotent. So the transition from a cell containing just the information to make one cell to a cell with two or more differentiated cells would have to occur, sharing tasks. Now let's suppose early Earth was populated by unicellular life forms like protozoans. Then the transformation to multicellular organisms began to evolve. Pluripotent cells would have had to become able to produce specialized cells, where each one of them would have had function only together with all other cells, like 10 different types of cells forming blood. This differentiation had to be organized and coordinated in advance, on a systems level above the individual cell level. Foresight is required to know the end function of blood in a joint venture of all required individual blood cell types. Blood is only functional if it carries all 10 different cell types, which have each different vital functions for life. Platelets, for example, are essential components of blood whose function is to stop bleeding by clumping and clotting blood vessel injuries. T cells are essential for human immunity. They orchestrate an immune response and play important roles in all arms of humanity. Red blood cells are the most common type of blood cells and essential for the vertebrate organisms delivering oxygen to the body tissues via blood flow through the circulatory system. At least 6 of the different cell types in the blood are life essential. If one is missing, life using cardiovascular systems is not possible. Had these blood types not to emerge together to make the life of a multicellular organism like vertebrates possible? Moreover, vascular cells have also been shown to be part of the niche that controls the maintenance and differentiation of hematopoietic stem cells, which form all blood cells and reside in the bone marrow. This forms an entire interdependent system where bones had to be existent, providing bone marrow the place where blood and vascular formation takes place. Most blood types require intermediate steps in their development until becoming final cell types used in blood. These intermediate cell types have no function in the organism. Why would evolution produce them or natural selection select them? We are talking about an enormous number of mutations required to produce each of the 10 different blood cells. Many cells play many roles during development, going through stages that are no longer seen in adulthood. How can natural selection account for the emergence of these cells? The role of some cell types is to activate specific genes in nearby cells, and once they accomplish that task, they die. This is clear evidence of orchestrated, pre-programmed processes. I think Byron Bledsoe brings it pretty well to the point in this quote. Irreducible complexity is powerful evidence of creation, and the vascular system composed of veins and blood cells and bone marrow, where hematopoietic stem cells are produced, is an interdependent system and so a very illustrative example. But how does science explain the origin of the blood vascular system? It is evident that the authors in the rather recent paper cited above from 2013, Evolutionary Origins of the Blood Vascular System and Endothelium, evolution is assumed, but the authors have nothing to offer besides declarations of vague guesses and assertions based on evolutionary beliefism. Morphogenesis. Morphogenesis depends on a series of factors, amongst it defining the size and shape of each cell, which has to organize into functional structures. Differentiated cells are not randomly distributed, rather, they are organized into intricate tissues and organs. During development, cells divide, migrate, and die. Tissues fold and separate. Genetic and epigenetic information has to be set up to define morphology, function, size and shape of each new cell, and how its new agglomeration leads to new functional structures like tissues and organs organized in non-random functional manner. Cells come in all sorts of forms and sizes, and that's defined based on the functions they will exercise. In an article in the magazine Wired, How Life Made the Leap from Single Cells to Multicellular Animals, the authors write, after the appearance of microbes, life got more complicated. Cells organized themselves into new three-dimensional structures. They began to divide up to the labor of life so that some tissues were in charge of moving around, while others managed eating and digesting. They developed new ways for cells to communicate and share resources. These complex multicellular creatures were the first animals, and they were a major success. Soon afterwards, roughly 540 million years ago, animal life erupted, diversifying into a kaleidoscope of forms in what's known as the Cambrian explosion. The longest-necked animals of all time were the extinct sauropod dinosaurs, some of which had necks 14 meters long. In these animals, the neurons that comprised the recurrent laryngeal nerve were at least 28 meters long. In the longest sauropods, these neurons may have been 40 to 50 meters long, probably the longest cells in the history of life. They were sensory neurons that connected receptors in the skin of the extremites with interneurons in the brainstem, a pattern of neural architecture that is present in all extant vertebrates. The biggest single-celled organism can be up to 20 centimeters across. One cell branches out into a network of tubes extending over 10 centimeters. As it grows, the deep-sea dwelling creature oozes slime onto the sediment, solidifying its structure. The size and form of a hospital are totally different than a residential house. What defines its structure is a scaffold structure, which has to be defined precisely taking the specific demands in consideration. Shape and form of cells have to be defined in accordance with the function that each cell type will perform. A neuron requires a totally different form and shape than a red blood cell. In cells, actin, ectomyosin, and microtubules that form the cytoskeleton exercise the same functions as a scaffold structure of a house or a large building, defining shape and form. For cells to function properly, they must organize themselves and interact mechanically with each other and with their environment. They have to be correctly shaped, physically robust, and properly structured internally. Many have to change their shape and move from place to place. All cells have to be able to rearrange their internal components as they grow, divide, and adapt to changing circumstances. These spatial and mechanical functions depend on this remarkable system of filaments. The cytoskeleton's varied functions depend on the behavior of three families of protein filaments: actin filaments, microtubules, and intermediate filaments. Microtubules are very important in a number of cellular processes. They are involved in maintaining the structure of the cell and provide a platform for intracellular macromolecular assemblies through dynein and kinesin motors. They are also involved in chromosome separation, mitosis and meiosis, and are the major constituents of mitotic spindles, which are used to pull apart eukaryotic chromosomes. The dynamic cytoskeletal and membrane system that governs morphological patterning in cells is fundamental. This system enables eukaryotes to package signaling molecules in endosomes, tiny vacuoles that bud from surface membranes, and transport them through the cell by means of molecular motors at rates much faster than would be possible by diffusion alone. As a result, eukaryotic cells can change shape or physiology in response to molecular signals, permitting processes such as locomotion of amoebas and permanent cell differentiation. The dynamic cytoskeleton and membrane system of eukaryotic cells opens up possibilities of size, structure, function, and development not available to prokaryotic organisms. Mitotic cell division is the most fundamental task of all living cells. Cells have the intricate and tightly regulated machinery to ensure that mitosis occurs with appropriate frequency and high fidelity. If someone wants to explain the origin of eukaryotic cells, the rise of mitosis and its mechanism, and the involved cell organelles and proteins must be elucidated. The centrosome plays a crucial role. It functions as the major microtubule organizing center and plays a vital role in guiding chromosome segregation during mitosis. These architecturally perfect structures are essential in many animal cells and plants, defining each cell-specific function. Genes define the amino acid sequence that makes proteins, and specific sets of proteins define tissue type. There are no proteins for hemoglobin in neuronal cells, and there aren't proteins that make voltage-gated ion channels in red blood cells. These fundamental elements of the cells, along with other cell-specific proteins, are responsible for making a neuron, and a red blood cell, a red blood cell. Yet both cell types have the genes for each of these cell-specific proteins. In fact, every eukaryotic cell with a nucleus in a multicellular organism contains a full set of all tissue-specific genes as part of the full genome. Tissue specificity must then arise from how genes are differentially expressed, meaning that in some cell lineages certain sets of genes are silenced, while in others they are expressed, leading one line of progenitor stem cells to differentiate into neurons and another to differentiate into erythrocytes, or any of the other approximately 200 different cell types of the human body. Another issue that has to be explained is individual and more advanced stage collective cell migration, which has a key role during morphogenesis and during wound healing and tissue renewal in the adult. It is a central process in the development and maintenance of multicellular organisms. In addition to displaying a coordinated migratory behavior, collectively migrating cells move more efficiently than if they migrated separately, which indicates that a cellular interplay occurs during collective cell migration. Yet in recent years, evidence has accumulated confirming the importance of such intercellular communication and the molecular mechanisms involved. The addition of cells in an evolutionary trajectory from unicellular to multicellularity requires the production of precise new information of where the new cells have to be located in the evolving organism. Processes such as tissue formation during embryonic development, wound healing, and the immune responses all require the orchestrated movement of cells in particular directions to specific locations. Errors during this process have serious consequences, including intellectual disability, vascular disease, tumor formation, and metastasis. Eukaryotic cell migration typically is far more complex and can consist of combinations of different migration mechanisms. It generally involves drastic changes in the cell shape, which are driven by the cytoskeleton, for instance, a series of contractions and expansions due to cytoplasmic displacement. When the tasks of labor are shared and/or divided in a multicellular organism, then the specific function to each cell must be purposefully defined, organized and assigned, in order to permit to build tissues, organs, organ systems to organisms. Epigenetics is a reference to a whole host of mechanisms that affect cellular function, tissue specificity, and morphogenesis above and beyond the genetic code specified in the linear sequence of DNA nucleotides. Each cell needs to be in the right position and place in the body. This is crucial. Limbs like legs, fins, eyes must all be placed at the right place. It is awe-inspiring to imagine that the instructional information stored in one single fertilized egg can give rise to a human adult body, developing into an organism made of 37 trillion cells, perfectly organized, each cell at the right place, emerging amongst 200 different cells, organizing into tissues and organs. Fingers are always at the tips of our hands, never in the middle. Our eyes are always in our heads, not in our toes or gut. This creation of ordered form involves precisely coordinated cell growth, cell migration, and cell death. To believe that such a complex organization would and could rise from unguided evolutionary accidental gene duplications, where the function of each cell differentiation makes only sense in the completion of an entire tissue or organ or organism, is irrational to the extreme. Even a trillion years would not be enough time to sort out the right development stages amongst all wrong ones. This conclusion is not an argument from incredulity. It is that evolutionary narratives stretch far beyond what is reasonable to be believed. I have not enough faith to believe that we are all the product of mere natural forces. Intelligent design by an unimaginably powerful intelligent creator is the only rational inference. Cell-to-cell adhesion is an essential feature of multicellularity. Of all the social interactions between cells in a multicellular organism, the most fundamental are those that hold the cells together. The apparatus of cell junctions and the extracellular matrix is critical for every aspect of organization, function, and dynamics of multicellular structures. During animal development, cells are typically free to migrate and slide past one another in ways that permit differential adhesion, cortical tension, and other processes that can facilitate the sorting and assembly of some tissues. The cells have not only to hold together, but important mechanisms to stick the cells together is required, that is, the ability to individual cells to associate in precise patterns to form tissues, organs, and organ systems. And it requires that the individual cells be able to recognize, adhere to, and communicate with each other. That is by no means a simple task. Even in a nematode worm, as many as 2,000 genes encode for cell adhesion and cell signaling. The other crucial innovation that had to occur and emerge is cell-cell signaling and communication. Even in bacteria and colonies do communicate with each other. That is by no means, however, comparable with the complexity of communication systems of multicellular organisms. Higher organisms have to coordinate a large number of physiological activities, cell-cell interactions through signal transduction pathways, and are crucial in the coordination of embryonic development. Signal transduction within the target cell must be coordinated, fine-tuned, and channeled within a network of intracellular signaling paths that finally trigger distinct biochemical reactions and thus determine the specific functions of a cell. Importantly, both intercellular and intracellular signaling are subjected to a regulatory mechanism that allows the coordination of cellular functions in a developmental and tissue-specific manner. Such a transition requires more than a mere augmentation of an existing system for coordinated multicellularity to evolve. It requires the ex nihilo creation of an entirely new system of organization to coordinate cell appropriately to form a multicellular individual. organisms have a variety of different mechanisms to achieve cell-to-cell communication. The mode of communication depends in part on the distance between the cells that need to communicate with each other. One way to categorize cell signaling is by the manner in which the signal is transmitted from one cell to another. Despite the bewildering number of cell types and patterns found in the animal kingdom, only a few signaling pathways are required to generate them. One clear conclusion to be drawn from all these studies is that there is a rather limited number of signaling pathways to generate remarkable variety and complexity to form and function that we see today, both across phyla and within the individuals of a given species. Signals are relayed between cells in different ways, all of which involve a cell that produces a signal and the target cell that receives the signal. Complex multicellular organisms, however, have an additional path for cell-cell communication in the form of microscopic passageways across cell walls and membranes. These passageways facilitate electrical, metabolic, and signal communication between cells and do so in especially specific or targeted ways. Since all signal transduction pathways are essential for multicellular development, is it feasible to suppose that they were co-opted and borrowed from unicellular organisms? If so, they still need the information to direct new body plan development. Where did this information come from? The rules of any communication system are always defined in advance by a process of deliberate choices. There must be a prearranged agreement between sender and receiver, otherwise communication is impossible. By definition, a communication system cannot evolve from something simpler because evolution itself requires communication to exist first. You can't make copies of a message without the message, and you can't create a message without first having a language. And before that, you need intent. A code is an abstract, immaterial, non-physical set of rules. There is no physical law that says ink on a piece of paper formed in the shape of TREE should correspond to that large leafy organism in your front yard. You cannot derive the local rules of a code from the laws of physics, because hard physical laws necessarily exclude choice. On the other hand, the coder decides whether one means on or off. She decides whether 0 means off or on. Codes, by definition, are freely chosen. The rules of the code come before all else. These rules of any language are chosen with the goal in mind, communication, which is always driven by intent. That being said, conscious beings can evolve a simple code into a more complex code, if they can communicate in the first place. But even simple grunts and hand motions between two humans who share no language still require communication to occur. Pointing to a table and making a sound that means table still requires someone to recognize what your pointing finger means. Most signal relay stations we know about were intelligently designed. Signal without recognition is meaningless. Communication implies a signaling convention, a coming together or agreement in advance that a given signal means or represents something. That SOS means send help. The transmitter and receiver can be made of non-sentient materials, but the functional purpose of the system always comes from a mind. The mind uses a material substance to perform an algorithm that is not itself the product of the materials or the blind forces acting on them. Signal sequences may be composed of mindless matter, but they are marks of a mind behind the intelligent design. Cellular interactions with the extracellular matrix and with neighboring cells profoundly influence a variety of signaling events, including those involved in mitogenesis, survival, and differentiation. Integrins, cateins, selectins, and other cell adhesion molecules regulate signal transduction cascades. Animal cells use specialized adhesion receptors to attach to one another. Many of these adhesion proteins are transmembrane proteins, which means the extracellular portion of these proteins can interact with the extracellular portion of similar proteins on the surface of a neighboring cell. All-Tau diagrams of adhesive structures may suggest that they are static once assembled. They are anything but. Cell-cell adhesion receptors fall into a relatively small number of classes. Cells can dynamically assemble and disassemble adhesions in response to a variety of events. This seems to be an essential requirement for function right from the beginning of multicellularity. Many adhesion proteins are continuously recycled. The molecular machines to exercise these functions therefore had to emerge together with adhesion proteins. The setup and implementation of sophisticated, complex, and advanced communication networks like the Internet depend on the invention of highly intelligent, skilled communication network engineers. Most impressive, multicellular organisms use several extremely advanced communication systems, like tunneling nanotubes, extracellular vesicles, which are on top of that also cargo carriers. The size of the communication and cargo delivery network of the human body is 75,000 times the size of the entire internet of the whole world. If there would be just one communication connection between each cell. In reality, things are far, far more complex. Each neuron cell computer is connected up to 10,000 other neurons. Cell-to-cell communication is a critical requirement to coordinate behaviors of the cells in a community and thereby achieve tissue homeostasis and conservation of the multicellular organism. Tunneling nanotubes as a cell-to-cell communication over long distance allow for bi- or unidirectional transfer of cellular components between cells. Health and performance are completely dependent upon how efficient that signaling and communication process works. Evidence suggests that these highly sophisticated communication channels had to be fully functional right from the beginning. Microevolution is better described as adaptation and is an engineered process, which does not happen by accident. The cell receives macroscopic signals from the environment and responds by adaptive, non-random mutations. The capacity of mammals and other multicellular organisms to adapt to changing environmental conditions is extraordinary. In order to effectively produce and secrete mature proteins, cellular mechanisms for monitoring the environment are essential. Exposure of cells to various environments causes accumulation of unfolded proteins and results in the activation of a well-orchestrated set of pathways during a phenomenon known as the unfolded protein response. Cells have powerful quality control networks that cooperate to monitor the folding states of proteins and to remove misfolded conformers through either refolding or degradation. Cell survival requires appropriate proportions of molecular oxygen and various antioxidants. Reactive products of oxygen, called reactive oxygen species, are amongst the most potent and omnipresent threats faced by cells. Cells damaged by ROS, irreversibly infected, functionless and/or potentially oncogenic cells are destined for persistent inactivation or elimination respectively. If mechanisms that do not trigger controlled and programmed cell death, apoptosis, are not present on day one, the organisms cannot survive and dies. The social collaboration within a multicellular organism is so close and intricate that it even extends to the self-sacrifice of the somatic cell for the sake of the germ cell and the perpetuation of the species. This is very different from unicellular organisms, where the principal rule is the survival of the fittest, or perhaps more accurately, the survival of the fit enough. Simply put, the principle is that all of a multicellular organism's cells are prepared to suicide when needed, for the benefit of the organism as a whole. They eliminate themselves in a very carefully programmed way so as to minimize damage to the larger organism. On average, the human adults it's about 50 to 70 billion cells that die per day. We shed 30,000 to 50,000 skin cells every minute. An adult human is made of up to something like 40 trillion cells, most of which are replaced on a time scale of months as they wear out or get damaged. So over a lifetime, a human might have to grow 20,000 trillion cells. Every one of those cells must behave according to the overall program. In multicellular organisms, genetic programs control precisely how many times each cell reproduces. But if those cells were allowed to reproduce uncontrollably in a multicellular organism, that would destroy organismal integrity and harm or kill the organism. At the organismal level, selection will favor traits that preserve organismal integrity, which tries to control reproduction of cells beyond what is needed. Organism seeks to control the reproduction to what is needed at a higher level of organization. A single cell seeks to reproduce more than its competitors. In a bacterial colony, if one bacterium can grow faster than the others, then the bacterium's offspring will eventually dominate the colony. That cannot be allowed to happen in a multicellular organism. If one cell gains a mutation that makes it grow faster, or where it is not meant to, the result is cancer that will kill the organism. If each cell in our face were to undergo just one more cell division, we would be considered horribly malformed. If each cell in our arms underwent just one more round of cell division, we would tie our shoelaces without bending over. How do our cells know when to stop dividing? Our arms are generally the same size on both sides of the body. How is cell division so tightly regulated? Body growth in animals is rapid in early life, but then progressively slows, thus imposing a limit of adult body size. This growth deceleration in mammals is caused by potent suppression of cell proliferation in multiple tissues. This progressive decline in proliferation results from a genetic program that occurs in multiple organs and involves the downregulation of a large set of growth-promoting genes. The limit on adult body size is imposed by a negative feedback loop. Different organs appear to use different types of information to precisely target their adult size. Organ size appears to be limited by initial number of progenitor cells, suggesting a mechanism based on cell cycle counting. growth disorders result in the unrestrained growth of cancer. Furthermore, other complex regulatory mechanisms that consist of multiple components with overlapping functions guarantee that defects in one component do not totally abolish the timing function. If some developmental systems' growth appears to be limited by a cell division counter. How came this to be? Designed programming by intelligence or set up by unguided evolutionary tinkering? The morphological shape of multicellular organisms is largely defined by the gene regulatory network. Cells must activate their genes at different times and places, which means complicated control systems of the genes called genetic control circuits. The more complex the organism, the more complex these circuits are. The gene regulatory network is essential for the development of the single-cell zygote into a full-fledged multicellular organism. Different body plans are realized with a set of developmental genes, namely transcription factors and signaling molecules. A network of interactions between the gene regulatory network and genes govern cell differentiation. The instructions that control when and where a gene is expressed are written in the sequence of DNA bases located in the regulatory region of the gene. These instructions are written in language that is often called gene regulatory code. These networks are known as developmental gene regulatory networks and consist largely of the functional linkage between developmental control genes, cis-regulatory modules, and differentiation genes, which generate spatially and temporally refined patterns of gene expression. Gene regulatory networks are modular and hierarchical in architecture, as they are divided into different components. For example, the components controlling the initial stages of development are at the top of the hierarchy, while the portions governing intermediate processes such as spatial subdivision and morphological patterns are in the middle, and the components controlling more specific functions including cell differentiation and organogenesis or morphogenesis are in the periphery. A cell is a sophisticated device that performs three elaborate functions: sensing inputs, processing the input information for decision-making, and executing the outputs. To this end, cells have built-in sensors that can receive the input signals generated by various environmental factors. Specifically, the plasma membrane and its integrated receptors can sense pressure, osmotic stress, intracellular contact, temperature, and chemicals. At the same time, reactive oxygen species, pH, nutrients, signaling factors, and other indicators of internal state are registered by internal receptors. Varying degrees of a single environmental input or a combination of many of them is presented to the cell at any given time, giving rise to a large array of input information sets. Cells continuously process this multitude of input signals to make decisions about their appropriate responses that lead to changes in gene expression, enzymatic activity, and rewiring of their signaling networks. 1. Regulation, governing, controlling, recruiting, interpretation, recognition, orchestrating, elaborating strategies, guiding, instruct are all tasks of the gene regulatory network. 2. Such activity can only be exercised if no intelligence is present, if the correct actions were pre-programmed by intelligence. 3. Therefore, most probably, the gene regulatory network was programmed by an intelligence agency. 1. Complex multicellular life forms depend on gene regulatory networks, which are a collection of molecular regulators that interact with each other and with other substances in the cell to orchestrate the expression of DNA. 2. Gene regulatory networks operate based on logic gates, and their networks process chemical input signals similar to computers. These encoded instructions are based on Boolean logic. 3. Logic depends on reason, and reason depends on intelligence. Only an intelligent mind can think rationally and implement a system based on conceptual laws of logic. Therefore, the best and most reasonable explanation for the existence of complex gene regulatory networks based on Boolean logic, essential for the making of complex multicellular organisms, is the creative action of a powerful, transcendent, intelligent creator. 1. The setup of functional information retrieval systems, like a library classification system, is always tracked back to intelligence. 2. The gene regulatory network is a fully automated, pre-programmed, ultra-complex gene information extraction system. 3. Therefore, its origin is best explained through intelligent setup. Epigenetic information is thought to be contained in cell structures other than DNA, and to involve, for example, patterns in the cytoskeleton, the cellular scaffolding or skeleton in a cell cytoplasm, and in the cell membrane, but these patterns too are probably effects of more fundamental causes. Darwinism, therefore, fails to account for the origin of both the genetic and non-genetic information necessary to produce new forms of life, and cannot explain what actually determines an organism's physical shape. From the year 2000 onwards, a new branch of biology, a concept called systems biology, has begun to be used widely in biology in a variety of contexts. Systems biology focuses on complex interactions in biological systems by applying a holistic perspective. Cells and organisms work based on data flow. Data processing can be found in nature all down to the atomic and molecular level. Examples are DNA information storage and the histone code. Moreover, cells have the potential to compute or process data both intracellular or transcription networks and during cell-to-cell communication. Higher order cell systems such as the immune and the endocrine system, the homeostasis system, and the nerve system can be described as computational systems. The most powerful biological computer we know is the human brain. The sperm and egg are highly specialized cells, and only they can transmit the instructions for making an organism from one generation to the next. How are these germ cells set apart, and what are the instructions in the nucleus and cytoplasm that allow them to form the next generation? The complexity and different systems needed to be in place for sexual reproduction to be possible are truly awe-inspiring and amazing. For good reasons, sex has been said to be the queen of problems for evolutionary biology, to explain its origin. Either the sperm can follow the female hormones to the egg, or they cannot. Either the sperm can penetrate the egg wall, or it cannot. If we all share a common ancestor, at some point reproduction had to evolve into the sexual realm, and because that has clearly happened, it must offer some kind of genetic advantage. But what is it? Science has no clues. In this video, I have given a short overview of the manifold innovations that had to occur to go from the unicellular to the multicellular realm. Of course, each of the 10 points mentioned deserves far more in-depth analysis and elucidation, but what has been shown outlines a main point of this video. There is nothing simple in biology. The transition to multicellularity would have required at least the 10 innovations at once. But that is not how evolution works, where slow, gradual mutations occur over long periods of time. The evidence in the fossil record demonstrates the appearance of multicellular organisms suddenly, like in the Cambrian explosion. And so does the evidence in the molecular world confirm that a huge transition to multicellularity had to emerge, with a totally new order and level of functional complexity, all at once. That is simply evidence of intelligent design and not evolution. Science magazine, for example, makes the claim because this transition supposedly occurred multiple times, it must have been a small hurdle. That's a remarkable non sequitur. What a misleading conclusion, which demonstrates how the evidence has to be twisted in order to fit the naturalistic narrative. No design inference permitted. I would say a nice example of the bad fruits of philosophical naturalism, which permeates modern science. If science papers claim that selecting just a few genetic mutations explains the enormous feat of increase of organized complexity, even if millions or hundreds of millions of years were at hand, then there are good reasons to be skeptic of such claims. A Above Science article is a little bit more courageous and admits that multiple innovations would have been required in these transitions, but does not explore where this evidence leads to further, but then simply sticks to the baseless claim that convergent evolution just happened. Lewontin brought it nicely to the point when he says, "Our willingness to accept scientific claims that are against common sense is the key to an understanding of the real struggle between science and the supernatural. We take the side of science in spite of the patent absurdity of some of its constructs, in spite of its failure to fulfill many of its extravagant promises of health and life, in spite of the tolerance of the scientific community for unsubstantiated just so stories, because we have a prior commitment, a commitment to materialism." Each of the topics mentioned in this video is huge, pinpointing what really defines body architecture, the orchestration of organism development, cell shape and body form, and the mechanisms of adaptation and first degree of speciation, and exposing the correct explanation of biochemical mechanisms of biodiversity is the holy grail of biological sciences. When science claims that evolution is a fact, it must be defined what is meant by it. There are still too many open questions in regards of macroscale evolution which have not been answered conclusively by science. Only who has no clue can claim that micro leads to macro. It does not. Pre-programmed codified information and signaling replaces Darwin's theory and its various subsequent adaptations, extensions, and new proposals like the modern extended synthesis or the so-called third way. The true mechanism is biochemical systems programming and signaling, and special creation of species and kinds by an intelligent, powerful creator. Long periods of time and gradual evolutionary development is not possible. Face the fact that cells and organisms work like gigantic interlocked machines and factory complexes, where in any case, if one tiny part is missing, nothing goes. Natural selection would not select for components of a complex system that would be useful only in the completion of that much larger system. I think the exposed requirements to form multicellular organisms, which is however just a tiny fraction of the trajectory from supposed last universal common ancestor to humans, this alone demonstrates Mount unsurmountable that evolution would have to be able to overcome. A watchmaker is required to make a watch. A blind watchmaker can't make a watch. Life is much more complex than any watch or any artifact made by man. Only upon what has been exposed in this video, I think it is very reasonable and rational to believe in a powerful, super intelligent creator which did set up life for his purposes.